It is my great pleasure to introduce Dr. Fiona Simpson from the Frazer Institute to talk to us today. Fiona may sound like she was born overseas, but she was born in Australia. And she did her PhD in Cambridge and then some postdocs in the United States before joining UQ at the IMB in David James's group. And she's worked a lot on trafficking, cellular trafficking, and after her time with Professor James, she set up her own lab at the Frazer Institute, where she focused a lot on EGFR. And she's now got a really burgeoning group and program around a couple of different cancers, head and neck, and also breast cancer. And we're very excited to hear her talk today. So thank you for joining us, Fiona.
Thanks, Amy, and hi, everybody. I'm just going to...
Fiona, are you on mute?
No. Can you hear me, Amy?
Oh, maybe just me that can't hear.
I can hear
Everyone
you.
else can. Yeah, we're good.
I'm not on mute.
All good. Thank you.
The people on Zoom can hear.
Yes, Fiona, we can.
Can you hear, Amy?
Yes, I can. Yeah.
All good. Okay. So I'll share my screen. So can everybody see that now? Okay.
Yep, looks good.
Excellent. Right. So I'd like to start with an acknowledgement of country. UQ acknowledges the traditional owners and their custodianship of the lands in which we meet. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country, and recognise the valuable contributions to Australia, global society and science, and welcome anybody from an Indigenous people's background who are joining us today. So if I can get this to move, wow. Okay, so these are just my disclosures. And what I thought I'd do today is show you a lot of our new work. So as we go through, you'll see a lot of the slides have unpublished data. They're marked with red. Please don't repost or image that data because a lot of it's under submission. So 2 years ago, the lab published a paper in Cell, and some of you might recognize Lorette, Florida, Priscilla, and Shannon, who were first authors on the paper and who did all the work. So these are the team. And how this started was actually in patients. And so as we go through, people kind of think that we started to move this therapy through the lab towards patients, but actually the whole project started with a signature in patients. And what we'd done was we were trying to look to see why 98% of head and neck cancer patients are EGF receptor positive. But in monotherapy, only about 15% of them respond to anti-EGF receptor antibody therapy like cetuximab, not at all to gefitinib, which is a small molecule inhibitor. And that goes up to about 30% when you add radiotherapy. It's synergistic. And so we wanted to know why these tumors that are so highly expressed in the receptor weren't responding to the antibody. And our idea was just to look at rates of uptake and internalization with the thinking that that might change outcomes. And we found two completely different patterns. It's actually in things like breast cancer, there's 3 patterns, but we were lucky to start with head and neck, and there's only 2. And it's very clear. Either the patient's tumors have slower EGF receptor endocytosis, so most of it appears to be on the plasma membrane, or it's internalized. And while our expectations were that surface level RTKs would be bad, it actually turned out after a couple of years when we matched up to patient outcomes that we had a 100% prediction of cetuximab response, but in the opposite direction than we expected. Where the receptors were slowed down on the surface, that's where we got tumor regression. Now, a lot of people get these images confused. These are not steady state images. You cannot see it by pathology. If you image the EGF receptor in these tumors by pathology, you see all the plasma membrane and all the internal EGF receptor. And it doesn't look like this. This is EGF with directly conjugated Alexa. And we did this in live human tumors within 15 minutes of removal in surgery. And what we did was old-fashioned biochemistry uptakes where we did 5 minutes, 10 minutes, 15 minutes in separate tumors, and then looked at the pattern or the rate of endocytosis. And so that hadn't been done before. And that's how we saw the pattern, which again, this is not level of EGF receptor. It's not the total distribution of the EGF receptor. So it's not steady state. It's a kinetic measurement. So I thought I'd make that clear because we really, as I go through the talk, you'll see that thinking in space and time and pharmacology applies to a lot of what we're doing. So we couldn't get that through to pathology as a prediction. It costs a lot of money and there weren't many companies willing to reduce their antibody sales. So what we thought we could do is turn the non-responders into responders. And that's how we started the whole project, because I trained in the States in Sandy Schmidt's lab, who actually discovered the function of this molecule, dynamin. So dynamin's a large GTPase. It wraps around the necks of clathrin-coated vesicles, and it's involved in fast endophyllin-mediated endocytosis, FEME. So these are the two pathways that the EGF receptor are internalized by, and they're both dynamin-mediated. So dynamin doesn't actually do the scission, it catalyzes the scission of these vesicles into the cell. So if you inhibit it, then things just can't get off the surface of the cells that are taken in by these pathways. So what we thought we could do is, if we inhibit it, dynamin in the patients, we could turn the non-responder pattern into the responder pattern. It was that simple. So we actually did that. We did it with prochlorperazine. Now that's used at low levels. You get it as Stematol as travel sickness tablets. At higher concentrations, it's an antipsychotic. At higher concentrations, it's used pre-surgery in some surgeries. And around those high surgical concentrations, slightly higher, it becomes a dynamin inhibitor. So at low concentrations, prochlorperazine will actually inhibit the dopamine receptor. And what that does is activate NK cells. However, what we've shown is that these NK cells get activated by dopamine receptor inhibition, but they've got nowhere to go. At the much higher levels of prochlorperazine, then dynamin's inhibited. And what you can see here is a kind of final, a picture paints 1,000 words. This is Rob Parton's EM, Rob's over IMB. And on the top here, you can see natural killer cells with their cytotoxic granules inside them. And they're contacting the tumor cells through these small synapses. And what we could see when we inhibited dynamin using prochlorperazine at these higher concentrations, you can see with the HRP label tag here, you can see the cetuximab bound to the EGF receptor stuck temporarily at the plasma membrane. Its endocytosis is delayed. And what you can see is that these natural killer cells form these really long synapses. And here you can actually see the cytotoxic granules being injected into the tumor cells. So the effect of this was not to increase the levels of the EGF receptor on the plasma membrane. The images that we show look like there's more, but it's actually just a pulling together, especially when we're using fluorescence of the fluorescent tags. And that increases what light we can capture with the confocal. So we've shown that we're not actually changing the levels of the target receptor on the plasma membrane. What we're doing is delaying their removal from the plasma membrane. Now, the effect of dynamin inhibition, because it doesn't work with clathrin inhibition, with dynamin inhibition, the receptors cluster together. That means the therapeutic antibody clusters together. And that clustering of the IgG tails is then enough to overcome the Fc gamma R receptor barrier of activation on the NK cells, and you start tumor killing. And we've been doing a lot of electron microscopy in vivo now, where we're actually giving therapies, taking the tumors out and looking by electron microscopy to look at the other immune cells that are involved in these very initial steps before the T cells come in. So this is a mechanism that we could use to, it doesn't improve highly sensitive cell killing, but it does help to reverse cells which are resistant to cetuximab. So we took that to clinical trial, and we've taken it through phase 1b, and the safety results show that this is safe. We saw some reduction in tumor masses, but this was a safety trial, not an efficacy trial. So the numbers aren't high enough to show efficacy. But what we did show was safety. And the other thing we saw is that in the cetuximab response, the patients developed a rash. And that rash, which is IL-17 driven, actually correlates with response to cetuximab. So one of the things that we noted through the trial is that 50 and 60% of the patients actually got a rash, as opposed to the monotherapy response rate of 15%. So we're taking that towards CSTEM2 in phase 2 now. And we've also started HER2-Pro, which I'll talk about. So this is kind of what I was talking about, where the green here is EGF bound to the EGF receptor, and it looks like we're increasing levels in the plasma membrane, but we're not. This again is a live assay. The EGF488 is used as a marker for cetuximab binding sites. And the reason that we see it better is it's directly conjugated. There's no secondary amplification. And as you cluster and bring the receptors together, that's when you see it. And we showed that we could see them using high resolution 3D SIM microscopy. So this is an effect of confocal, and what we're actually seeing is the clustering of the EGF receptor. So we've started HER2-Pro. There's 2 sites, one here in Brisbane and one with LG and Lim down at St Vincent's in the Garvan in Sydney. And that's basically to add prochlorperazine to the dose studies with paclitaxel, trastuzumab and pertuzumab. And we're treating in HER2-positive metastatic breast cancer. And that's ongoing at the moment. And Kate Cuff is the treating clinician and lead for that side of HER2-Pro here in Brisbane. So I wanted to talk to you about the new work and where this has all gone in the last 2 years. So one of the things we noted is that this delaying of receptors and increase in antibody-dependent cellular cytotoxicity also works for the checkpoint inhibitors. And one of the issues with the checkpoint inhibitors is it's really hard to see whether you're getting response or whether the tumor increases from progression or immune cells going in. And we were getting really fascinated, especially with electron microscopy, about how that immune cell, tumor cell synapse is controlled and formed. Now, in immunology, there's lots of signals which activate immune cells. And we know the end result of that activation, but all the molecular pathways in between are not characterized. So one of the things we've been working on are these sets of proteins, which actually, they start around the centrosome and they're responsible for the biogenesis of the immune synapse. So those immune proteins that form the immune synapse when the cell is activated are actually moved out from inside the cell to form the immune synapse. It's the same way a neural synapse would be formed. And then after activation, all those components are endocytosed to form the next set of immune synapse or cytotoxic granules. So in some cases, immune exhaustion is not actually that the cells are exhausted, it's just that they've cleared their plasma membranes and they're in the point of processing to remake these in the same way as neural synaptic progression occurs. So the red protein here, which I won't be naming, is a protein I actually identified in the States. And it's a family of proteins that are involved in moving molecules around the cell. And it's, in this case, this particular homologue is involved in NK cells, B cells, and T cells. So we've been characterizing that whole complex. We've identified the motors, we've identified the signaling responsive molecules, and we're hoping to basically fill in the entire black box that occurs between immune cell activation and immune synapse formation. The nice thing about this marker is that if the immune cells activate in response to antibody therapy, we actually see it relocalizing from the microtubule organizing center to the plasma membrane as those immune synapses are formed. So we're talking to people about getting validation cohorts, because what we're hoping to do is use this as a pathology marker to show whether patients' tumor and immune cells actually not only do the immune cells activate in response to those antibodies.
Yeah, you're right. Yeah, I am.
So not only would they activate in response to those antibodies, but also we actually see an active immune synapse killing synapse form, which is different from them just being there. The other odd thing about some of these proteins is in healthy cells, they're absolutely immune specific. But in some cancer cells, particularly in some types of breast cancer, these are being expressed. And so we've looked at that in the cancer cells. And what happens is that if you express parts of these machinery in cancer cells, they start to co-express immune checkpoint inhibitor receptors like PD-L1, CD20, which is the FCR. And those can form a pseudoimmune synapse. And so we're actually looking to see if the expression of these immune proteins, which shouldn't be expressed in the cancer cells but are, is actually forming these pseudoimmune synapses and moving all those checkpoint markers. They're actually causing them to be expressed. They upregulate them. If you transfect some of these proteins into cancer cells, they're suddenly expressing checkpoint inhibitors, receptors all over, and different kinds. And so one of the ways we thought we could deal with the heterogeneity of the checkpoint receptors that are expressed in immune cells is if we can knock out this machinery, then what we're hoping to do is actually remove the immune privilege from cancer cells, and then our immune system should clear it. But that's a long-term goal, obviously. We're talking to the chemists about drugging some of these molecules at the moment. On a kind of more straightforward front with our technology, we've been looking at antibody-drug conjugates. Now, it's actually been published in a paper. People's reasoning is that you can't use inhibition of endocytosis to improve ADCs because antibody-drug conjugates, where the toxin is bound to the end of the antibody, actually require delivery inside the cell for the linker to be cleaved and the toxin to be released and be active in the tumor. However, the thing with drugs is that they're reversible. And so what you can see here is the surface of a tumor taken from in vivo time points. And when we're using prochlorperazine as a dynamin inhibitor, the dynamin inhibitory concentrations only last from between 2 to 4 hours, depending on which receptor. And at the point of reversal, pharmacological reversal, all the antibodies built up on the surface of the cells then internalize together. So please don't put this out. This I've just written with the lab invited review for Nature Cancer Reviews. This is one of the figures. It's with the artists and publishers just now, so please don't post it anywhere. But what you can see here is that if you delay endocytosis, then you can get increased activation of the FCR. You allow more time for drugs in the plasma and the blood to bind to the receptors before they're internalized into the cells. You get everything loaded up, and then as endocytosis reverses, you get a wave of delivery into the cells. So we gave our dosing protocols and prochlorperazine to John Hooper's lab, and Thomas Frazer and John and his guys did these experiments in their pancreatic cancer models, with their anti-pancreatic cancer antibody, and they use prochlorperazine. And what you can see here, again, is that we get a marked improvement when prochlorperazine is used in combination with their ADC. So that was kind of an in vivo proof of the conceptual drug delivery that we had. So we've been working on that and delivery of radioligand therapy and improvement of radioligand, just radio imaging, PET imaging. An odd side effect. So in the Cell paper, we showed that it was absolutely the delay in endocytosis that gave us the therapeutic effect. And if you use identical IgG1 chain therapeutic antibodies, which are not internalized by dynamin-mediated pathways, you get no therapeutic effect at all. It's absolutely the endocytosis. However, PCZ is a multi-target drug. It activates immune cells through dopamine receptor inhibition. It improves ADCC through dynamin inhibition. And one of the other side effects we discovered in, especially through the human trials, is, and people say this isn't very scientific, but the surgeons know what I mean. It changes crunchy tumors to squishy tumors. Now, that's really important, especially in things like PDAC and head and neck cancer, where they secrete huge amounts of collagen. And one of the issues with that is that access for immune cells and drugs becomes limited, and they become immune cold environments. And so one of the side effects of the doses of prochlorperazine we're using is it actually cross-links collagen. And so we're just setting up to work with a Danish company whose drugs are developed to do this, on finding out exactly what PCZ's target is here and making specific drugs to be able to do that. So we've applied a lot of these to new models where we work on some rare cancers. We work on perineural invasion in the head and neck region. So this is where people have skin cancer like cutaneous SCC in the head and neck area. And what appears to happen is that it contacts the nerve endings, and some of these tumor cells then migrate up. So there can be patients who've had their primary tumors removed, they've been told they've got clear margins, and then 6 months to a year later, they get a feeling of, it's called formication, like ants crawling up their face, or their face starts to have some paralysis. And that's actually a sign of this perineural invasion, where the cancer's invaded the nerve. Now, at the moment, that's treated by surgery and RT. If the surgeons can't remove enough of it, it keeps tracking up the trigeminal nerve and it kills the patients. So we did some simple pathology, first of all. So head and neck SCC is like over 90%, 98% EGF receptor positive. It's different in cutaneous SCC. Cutaneous SCC skin cancer is only about 45% EGF receptor positive. But when we looked at perineural invasion, it turned out it's 90% EGF receptor positive. So we thought, well, is it getting that receptor expression as the tumor cells go up the nerve, or is it already there in the primaries? So we went back to all the primary tumors from those PNI patients through pathology, and we basically found that they're the EGF receptor positive ones. So this is a result I just presented in Florida at the North American Base of Skull Surgeons Conference, because with PNI, what that says is that if your initial cutaneous SCC was EGF receptor positive, those are the patients that are more likely to develop perineural invasion. So we set up clinical testing models for this with Glenn Boyle at QIMR. He gave us his whisker pad models, which is like the nicest collaboration move ever. And we're using the sciatic nerve model from New York from Sylvie DeBoard and the folks at MSC. The nerve invasion, the sciatic nerve model has the advantage of it's 100% penetrant, so it's good for drug testing. And the whisker pad model is not fully penetrant, but it's really good for looking at it in the context of the area that PNI actually forms. So Priscilla Olivia de Lima had, once she published the Cell paper, this is the work that she went on to do because she's stunning. She's just finishing this up now. And she went through a number of different treatments to see if we could improve PNI. So what we could see is that most of the PNI was responsive to cetuximab, and that improved with PCZ. Avelumab, which is an anti-PD-L1 antibody, didn't really have an effect here, even in the triple combination. And in fact, what we find now in 3 different models is that if you add targeted therapies like Herceptin or cetuximab at the same time as checkpoint therapies like anti-PD-1 or anti-PD-L1, I'll show you sepilimumab later, that actually has a negative effect on efficacy. So Riccardo Dolcetti's also noted this down at the Peter Mac, and what appears to be happening is that if you give the checkpoint inhibitors too early, then they stop the ligand-induced effects of cetuximab or trastuzumab. So that's really interesting because there's a lot of these combination clinical trials happening just now, and we're kind of wondering what preclinical data that was based off, possibly not fully humanized mice. But we're all just about to put all that data together and put that out, and it talks to the staging of these drugs. Like if somebody's going to be potentially cetuximab responsive, then you actually need to get that activity started before you start with the checkpoint inhibitors. Excuse me. The other thing with this is, even where we managed to completely clear 5 out of the 6 PNIs, at the pathological level, inside the nerve, further up from the main perineural tumor, even with complete clearance, we could still see some cancer cells left inside that nerve. So one of the messages to this for the surgeons at the conference was, if you can cut a little further without further damaging your patient, then take as much nerve as you can. But also, there's a beautiful new paper out from Sylvie Debord in Cancer Discovery. And the videos in this are amazing. What's happening in the nerve is that Schwann cells are coming down, expressing galectin, and they form tubes. And they actually attract the SCC cells up these columnar nerves. Now, some of you might not know, but EGF receptors are receptor tyrosine kinase in adults. But during development, it's actually neural outgrowth and cytokinetic factor in the brain. And so it appears that that's why the EGF receptor positive SCCs are those that are finding the nerve endings and tracking up with the Schwann cell attraction. So we're all working at the moment to try and disrupt that interaction, which will allow us to fully clear perineural invasion. We also, because we're in head and neck work on adenoid cystic carcinoma, James Nightingale, who was a surgeon working in the lab on his PhD, just completed his PhD. He's now a doctor. And we've done the pathology to match a new classification that's been proposed in the literature for adenoid cystic carcinoma. They proposed the new classification whereby some are P63 and some are EGF receptor positive based on genetics. And we've just gone through all our pathology and confirmed that that's exactly right. And we've also been able to show from that that it influences patient outcomes. And so we'll be, that's in Cancer Discovery Clinical Cancer Research. So we'll be publishing the matching paper to show that their prediction's right, and it's actually predictive of patient response. In adenoid cystic carcinoma, it's actually better if you're EGF receptor positive than P63 positive. So with working on the radioligand therapies, we've also been looking at PET uptake, and Ben Panizza and his team at PA Hospital have now swapped to doing PSMA PET for their adenoid cystic carcinoma patients, because PSMA is not prostate specific, it's expressed in a number of cancers. And what's been shown is that PSMA PET with gallium imaging actually allows better imaging than the standard. And so all our adenoid cystic patients through our MDT are now being run through this because it's easier to see the spread and the metastasis, and it allows the surgeons better surgical planning before they go in to remove those. We tried using adenoid cystic cell lines They were a nightmare to get hold of in the middle of COVID. We had couriers running out to Boston Common and various other places to meet people with all the restrictions. But it turned out that none of the cell lines in adenoid cystic carcinoma reflect actual patient adenoid cystic carcinoma. So the Global Adenoid Cystic Foundation have been very kind. Normally they test other people's therapies with their PDX collection, but they don't fully humanize their mice the way that we do. And so they've given us their PDX collection for use. And what you can see here is part of Benedict Lum's PhD that he's submitting today, like in an hour. And again, looking at Cetux, PCZ, Sebi, and the different combinations. So once again, we can get improvement using prochlorperazine as an adjuvant for the antibodies. And again, as with the PNI models, we saw that you really need to stage Cetux and Sebi, the same as we did with Cetux and avelumab. There's just a timing issue there to make these combinations work well. So that's quite a short talk today, but I wanted to give you an overview of the different projects that we're doing because we love collaborating with everybody. We're funded by Rotary. We've been funded in the past by NBCF, Queensland Head and Neck, Project Pink, PA. I'm currently NHMRC funded for the first time in 20 years of application. And that's kind of nice. It's a weird position to be in. I work with terrible numbers of clinicians. And we're also working with David Cavalucci across at RBWH. So we're starting to spread across the river. Apparently the Brisbane River is crossable. And these are all my acknowledgements. Thanks.
Thanks so much for that, Fiona. Just if you listen really closely, you can hear all the applause coming from the auditorium.
It's so weird. I've been in-person meetings for ages, and it's really weird giving a Zoom again.
Well, look, we've had 40 people listening to you, so there's a few here. Everyone, feel free to put your questions in the chat or just raise your hands if you'd like to ask one. I guess I'll start. Fiona, you presented a number of different kind of clinically relevant studies there. Which of your projects do you think is closest to implementation in the clinic?
Well, I guess that's a mix with the clinical trials. It's either HER2-Pro, where we're in the middle of a safety lead into that. If that's successful, then Elgene plans to treat 468 patients. And so CSTEM2 got a little delayed here for various reasons, but I've been in the States a lot, and the MD Anderson are going to help us take that through because they have oncologist choice. Yeah. And they treat a lot of their PNI and head and neck patients with cetuximab. So it's not difficult for them to add a $5 bag of PCZ. And they actually get paid, the American clinicians, for every IV bag they put up. So they're quite keen. So we've had some issues getting this done in Australia, so we might be able to do this through the MD Anderson instead. We've been trying to keep everything Australian, but you know, MD Anderson. So those are probably the closest. The other things are always work in progress. I think what I'm most excited about at the moment is the immunology that we're discovering, because we're doing that at the molecular and cellular level. And if we can work that out and how to drug it, then if you want to modify immune responses, either autoimmune disease or in cancer where you want to increase it, we could actually alter immunity using drugs instead of having to ablate patients' immune cells to replace them. So then all those therapies for things like asthma and Crohn's disease and lupus become, if you've got the control mechanism and you can play with it pharmaceutics. So we're really excited about that because that's nowhere, and we've just accidentally kind of stumbled into the whole mechanism.
True serendipity, hey? Does anyone else have any questions? Very quiet group today.
Sunil had one.
I think he was just clapping you.
HER2
I was
tends
just
to get-
clapping you, saying it was a great talk.
HER2 tends to get recycled back to surface. Does PCZ change the internalization fate of the receptors? So prochlorperazine at a dynamin inhibitory level slows down HER2 internalization as well. A lot of people thought, I guess logically, the thinking was if you stop endocytosis, then as everything goes along, you'd actually increase surface levels of receptors because of exocytosis. That's not true. Endocytosis and exocytosis are in complete balance because it's not just proteins that get trafficked, it's lipids as well and fluid. And so if endocytosis is halted, you have to halt exocytosis. Otherwise, you keep adding lipid further and further to your cell and you basically burst it and vice versa. And so whenever one of those pathways is inhibited, the other automatically shuts down in the cell, and it's actually controlled by osmotic pressure and osmotic balance. So if we temporarily hold those receptors in the surface, one of the reasons we're not getting a big increase in levels in the surface is exocytosis is stopped too. Sunil.
No, I wasn't asking a question. I was just clapping, saying it was a great talk.
Yeah.
I've heard you before, and it's very nice work.
Yeah, it's a lot of that stuff now. It's unpublished stuff that we've done in the models, but the stories weren't as simple outcomes as we thought. So like the triple combinations, like everybody has these trials of target therapies in with checkpoint therapies, and they've all put them in together. And I think they've gotten away with that because all these drugs are in clinical practice and they're just adding them. But if any of them had done preclinical models, and I've talked to a couple of people doing this now, like Ricardo, they'd have seen that it's actually bad. Like it decreases the efficacy of both therapies. You have to stage them. So, you know, that's led us to some complications, but we really need to write that up and get it out because so many people are on these trials.
Yeah, definitely, Fiona. I know in the breast cancer space, there's a lot more effort being put into addressing sequencing of these combinations. But maybe if there was some firm preclinical data, people would be more inclined to impact it.
It's coming out. So before the surgery conference, I was at the Hallmarks of Cancer conference in San Diego for Cell. And there's at least 4 groups about to publish on the staging of surgery and checkpoint inhibitors. Because surgery, if you give checkpoint inhibitors like within 3 months after surgery, you can actually drop the efficacy.
Wow.
So the staging in all these things to get towards maximal efficacy is going to be really important. But there's a lot of data coming out in that now. And surgery really, really kind of suppresses immune stuff in odd ways. And one of the things we want to look at with the MD Anderson, because it's their radio oncologist as well, is one of the things that radiotherapy seems to do is it might actually delay, as with PCZ, the endocytosis of these things into the cells. And a lot of people are looking at the pharmacology because antibody clearance, a lot of it is actually endocytosis. And so if you're trying to target a cell surface receptor and the cell immediately internalizes it and degrades it in the lysosomes, then your cells are resistant. So there's a lot of work going on maximizing the killing effects and the bystander effects before the target receptors get destroyed as well. So it's a lot of thinking in space and time and movement. It's nothing stationary. It's a lot of fun for a trafficking person.
I think that concept that you're describing is actually permeated quite a lot to the clinical space, perhaps more than you realize, because the last few clinical trials meetings I've been to, there's been a lot of talk about the order in which even the standard drugs are used. Not just new drugs, but and thinking about whether the order of them are correct and whether they could be made more efficacious, not by adding new ones, but by simply switching the way that you use them. So I think people are getting the idea that this is a dynamic process. It's adapting all the time, and therefore you need to be aware of that in terms of how you give your therapies.
And I think we've been so far down the road of developing new drugs, right? Like people are looking for these smaller and smaller receptor numbers in cancer cells to try and target them. And my thinking is like head and neck cancer is a great example. 98% of those express EGF receptor. We have good drugs. Why are they not working? Can we make them work? And like I've been looking at adenoid cystic carcinoma, and something that really stood out at the surgeons conference was somebody gave a summary. And there is about 50 drugs that should have had an effect in adenoid cystic. And the most any of them do, including Cetux, is stabilize the tumor. So with Rob Barton, one of the things that we see in the very first stages, and we saw this 10 years ago in our head and neck patients, we did electron microscopy of responders versus non-responders. And we saw eosinophils. And the immunologists here at the time told me that they were the mop-up crew and they didn't do anything. So there's a beautiful paper just in the last 2 weeks in Cancer Cell showing that the very first response to checkpoint inhibitors is eosinophils. And in the EM we've been doing with Rob and all these models, what we can see is eosinophils. Now, if you look at adenoid cystic carcinoma, one of its molecular features is as it develops and as it advances into later stage disease, it loses all its eosinophils. But there's a disease in patients where their salivary glands in an allergic response fill up with eosinophils. So we thought, well, why are we inventing new drugs for adenoid cystic? Because the best options have already been tried. What we need to do is get the eosinophils into the adenoid cystic tumor so that the drugs we've already invented work. So we're just looking at ways with Ranjeni and Gabrielle and people of getting like IL-5 expression or eosinophil attractants directly injected into ACC, and then try those drugs again and see if they can finally work. You know, I really think in tumors, we're at the point of it's not so much new drugs as we need to make the ones we've got work better.
Absolutely. Fiona, there's another question here asking, is there a difference in treatment outcome between pre-treating with PCZ first before her targeting versus administering both together?
Yeah, the PCZ has to be later. There's a staging thing there too. So what we found, first of all, is we always get the patients on the antibodies first because the first 3 days of delivery it spends in their liver. And they don't reach plasma stabilization for at least a week. Cetuximab is quite good because it's stable in serum for 3 weeks. That's its half-life, it's great. And then you can add the PCZ just to cycle things. So we do that first because we have to work out that the patients aren't allergic to the antibody drugs. So adding PCZ on top of an allergy might not be a good idea. So we wait 1 or 2 weeks. And then the other thing about that is we found through all our ADCC assays is that if you inhibit all the endocytosis in the immune cells first, they can't actually form those immune synapses because they have to traffic too. And so what we do is we kind of get everything lined up and then we add the PCZ and that increases the killing. So our sequencing is always get the antibodies in there, get them to stable plasma levels in the patients, and then the PCZ is given a cycle. And the easy way to do it is to give them a 20-minute rest after their antibody and then put the PCZ up, and then they don't have to come in for extra hospital visits. It's not a big extra thing for them. Yes. We've been talking to a couple of people because the nice thing about PCZ is it's an antipsychotic and we absolutely know it's brain permeable. And we're also working with Rod Minchin and Lisa Kamanskis, who are looking at liposomal delivery and various pharmacokinetics of PCZ for us because they're like real pharmacologists. So, yeah.
Well, that's great, Fiona. It sounds like you've got a lot of exciting things in the pipeline. So hopefully we can get you back in the not too distant future and you can update us on where all these things are going.
I'm just lucky I've got a lot of great people in the lab. So maybe we should get them chatting about their work.
Excellent. So thanks very much for joining us, Fiona. If anyone wants to reach out with either samples or questions, I'm sure Fiona would be happy to receive your emails.
Lovely.
Thanks, everyone.
See you again. Bye.
Thanks,